On this episode of Pets Explain Science, we welcome the CRISPR crew. In 1992, Spanish molecular biologist Fransisco Mojica and his colleagues made an interesting discovery. Mojica was working with microorganisms that require high salt conditions in order to survive, and Mojica wanted to understand how they were able to grow in such high salinity. He found that their DNA possessed a series of regularly spaced repeats, or tandem repeats, called treps. He also found these in other prokaryotes in 1993.
Mojica hypothesized that the repeats were connected to the segregation of chromosomes in cell division. He included this hypothesis in a paper in 1995.
However, they were greeted with a backlash from the scientific community. Mojica even turned to the Spanish government for financial support, but was refused as a number of complaints had been issued regarding his work.
But in 2000, Mojica and Ruud Jansen, another biologist, teamed up to search for repeats in prokaryotic organisms.
Jansen discovered genes next to the repeats, and in 2002, he and Mojica came up with the name CRISPR.
What kind of name is CRISPR? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Mojica also discovered that there were sequences between the CRISPR repeats that he called the spacer regions.
The sequence in these regions matched the sequence of the particular virus, and Mojica realized that these DNA sequences protected the prokaryotes from being infected by viruses carrying the same sequences in its genome.
Basically, this gave the organism with CRISPR repeats an adaptive immune system.
This is why those prokaryotes were able to survive in conditions with high salinity.
Oh, and then, in 2015, CRISPR was declared Breakthrough of the Year by the American Association for the Advancement of Science. Pretty cool, huh?
Wait. Hold on a second. This all sounds really cool and all, but what does CRISPR do?
CRISPR is a natural gene editing system found in human biological defenses. It also lets the bacteria identify and destroy foreign DNA in the body.
Oh yeah, and it's better, cheaper, and faster than other gene editing systems. Yes, and Cas9 was also developed along with CRISPR, an enzyme guided by RNA that targets the DNA sequences to be cut out. This can happen by deletion, disruption, and correction or insertion.
Another cool thing about CRISPR is that it has been used in China successfully on human embryos to remove genetic defects. However, the use of CRISPR is still under consideration when it comes to use in the US due to safety concerns.
And guess what else? CRISPR can be used in plants and animals, just like us.
CRISPR may very well provide the means by which we can edit and repair defective and potentially dangerous genes. Well, folks, that's it. We'll see you the next time you need real science-y info.
With all the love from our fuzz, the CRISPR crew.